Evening, everybody, and welcome to the 10th season of Basic Science Lights the Way. Over the last 10 seasons of this show, we've been exposed to the brilliant stories of research done by our faculty, postdocs, and students. Tonight, we're going to hear some exciting stories about lungs and breathing. Breathing, one of my favorite activities. And so we'll hear from Ahmad Nabhan. First of all, he's going to be discussing one of my personal favorite topics, that of Wnt signaling, but essentially he'll be discussing how one kind of signal is interpreted in the lungs in two very different ways. One is good, which promotes regeneration and proliferation of lung tissue, and the other is bad. It promotes fibrosis and scarring. So he'll talk to us about his insights and where that research may lead in the future. Our second speaker is Sarah Stanley, coming freshly from her award of the Distinguished Teaching Award. So congratulations, Sarah. Great news to hear that you won this distinguished award. She's going to be talking about pathogens, tuberculosis, and how that is regulated by the immune system, and particularly how cells can either destroy or fail to destroy these pathogens. And let's see, I made a mess of my notes here. The third speaker is Diana Bautista, and she'll be talking about how some pathogens hijack the sensory cells of the lungs and cause the coughing and sneezing that spreads the disease. So this is a fascinating story, and we look forward to hearing that too. So now over to our moderator, Andy Dillin. Andy, thank you.
Thank you, Richard. Yes, okay, I'm unmuted now. Okay. Thank you. I'm very happy to be here. This is a really fascinating topic to be discussing, lung health and disease. And I am not new to this. So while my work focuses on whole body physiology and aging, early in my career, I helped develop a treatment for cystic fibrosis that is based on the activating cellular response system to repair the ion channels in people that had this disease. So we now appreciate that there is an extensive set of networks to manage protein environments and help cells deal with their diseases. In this case, such diseases as cystic fibrosis, this network fails to work as it should. And so with this new development, we're able to map this new network, leading to breakthroughs in understanding how this network, including aging, impacts environmental and cellular stresses, can lead to the onset of multiple serious diseases. Tonight, I'm really excited, looking forward to revisiting this fascinating organ, the lung, and hearing from some of my colleagues tonight about the latest advancements in lung research. Our first speaker this evening is Assistant Professor of Cell Biology, Development, and Physiology, Ahmad Nabhan. Ahmad joined the Berkeley faculty just recently in January of this year. He's getting his lab set up and running. And his research is focused on decoding and recoding stem cell communications and regeneration of fibrosis. That may sound like a mouthful, but Ahmad's going to make it very clear to us exactly how these cells are actually communicating with each other and helping the lung actually form and be functional. His lab's central focus is on molecularly decoding stem cell niche communication to better understand tissue maintenance and ultimately to reprogram this crosstalk for precision therapies that enhance organ repair. And we're going to hear about some of those precision therapies. They use lung alveolar stem cells as a model to study these broad questions. And these stem cells are amongst the most dangerous cells in the body, and disruption of their function underlies development of lung cancer. Ahmad, please tell us about your work.
Thank you, Andrew. And I'm just going to share my slides here. Okay, that's apparent to everybody. Okay, great. Thank you. And I'm really happy to be here, and I can't tell you how excited I am to be part of the Berkeley community. Unfortunately, I have to start this talk with a bit of bad news, which is that we as humans have a really limited capacity to heal our organs. So for example, if you have one episode of acute kidney injury early in your life, you're 10 times more likely to develop chronic kidney disease and require dialysis later in life. Similarly, if you have one bad lung infection, you're twice as likely to die from a respiratory related illness. really underlying that our capacity to repair these organs is limited. Now, yes. So on the other hand, there are these other organisms out there that you have probably heard of. And so these are organisms or animals like the axolotl, which have no problem regenerating at all. So you can essentially chop off its limb. You can also chop off its lung here, and both of these organs grow, sort of underlying this difference. And what's really amazing to me when I read these papers is that these animals often achieve these miraculous feats of regeneration without magical genes or exotic pathways, but by using the same genetic components that are found in our genome. And specifically, I'll focus today on these components that are used in cellular communication. And what I want to tell you is that if we can learn how our cells communicate, we can then learn how to talk back to these cells and change our intrinsic capacity to heal and copy some of the things that are going on in the axolotl. And so I'll be telling you about this today in the context of the distalmost compartment of the lung, which is called the alveolus. Looks like this. And here there are these incredibly thin, beautiful cells which do the gas exchange. And often these cells are lost due to normal wear and tear. And the stem cell, which is shown here in red, is able to self-renew and basically replace the lost cell with help from its supporting cast, which is a fibroblast, shown here in gray. However, a variety of problems like mutations, viruses like COVID, or even chemotherapy can, in some cases, compromise the capacity of the stem cell to proliferate, leaving behind these gaps in the epithelium. And so what happens then is that these gaps persist, where you're supposed to have gas exchange, and the fibroblast kind of freaks out and invades the space and fills it with scar tissue, ultimately causing organ failure. This disease is known as pulmonary fibrosis, where the patient is slowly choking from the inside out. This is a terrible disease with no curative therapy, really, other than lung transplant, which itself is not a trivial option. And essentially, once people understood that these two processes were at the center of the disease, the race was on to figure out, A, what controls stem cell function and how can we boost it? And B, what controls fibroblast scar formation? And can we perhaps slow it down to give these defunct stem cells some time to do their job? And as a graduate student, what I had actually discovered was that Wnt, which are a family of cell communication proteins, control the stem cell capacity of these alveolar stem cells and can even rehabilitate dysfunctional stem cells, which was really exciting at the time. It was sort of highlighted this idea that you can add an exogenous or an outside protein into your body and it can repair or rehabilitate your cells. However, there was another group of biologists at the same time which studied the second step of the process, the scar formation. And what they have found was that Wnt really promotes the ability of this fibroblast to make scar tissue, and adding extra Wnt not only would boost the stem cell, but it would also boost the scarring. And so you have this strange situation where two groups of people wanted to study the same disease by activating and inhibiting the same set of molecules. And in a sense, both groups are right. Really, the key to harnessing the therapeutic potential of these Wnt molecules is decoupling their effect on regeneration and scar formation. And we didn't really know how to do this at the time, but what we did know was that our organs had figured it out. So Wnt is often selectively delivered and selectively activates different cells within organs during development. And so perhaps if we can understand how the body does this, we can sort of harness it in a therapeutic setting. That was our idea. So the key hint really was that Wnt are a large family of proteins. There's not one Wnt molecule, but there are 19 Wnt ligands in our genome, and they can talk to any of 10 frizzled receptors, which are found on the surface of the cell. And so it seemed to me like this complexity would create a code-like mechanism where the different Wnt's and different frizzleds can selectively talk to one another, similar to how you can listen to a radio on different channels depending on the frequency. And so this was a simple idea, but technically it was very challenging because there are, again, 19 ligands, 10 receptors, and about 50 cell types in the lung. So mapping all this complexity was a daunting task. However, we used this technique called single-cell RNA sequencing, where hundreds of thousands of cells are captured in this manner that you see here. And in these tiny bubbles, we're able to analyze the presence of these various components. And this revealed really a very simple pattern, which was that fibroblasts and stem cells, which both rely on Wnt, actually express different receptors, as you can see here. And so the stem cell expresses Frizzled-5, and the fibroblasts predominantly express Frizzled-1. So if we could selectively activate Frizzled-5 without touching Frizzled-1, then we can achieve our goal of promoting repair while circumventing the effect on fibroblast scarring. Unfortunately, activation of Frizzled-5 with a Wnt ligand is actually not possible because Wnts are biochemically very difficult to work with. So we cannot really use what nature has created. However, now that we know the basic biology that we're trying to target and we've learned it, we can actually engineer a Wnt mimetic. So as you can see here, this doesn't look like a Wnt. It doesn't really smell like a Wnt, but it can be engineered to do the same job as Wnt, and it can be engineered to selectively interact with any of the 10 frizzles with a high level of potency and specificity. So that's what we did, and we wanted to test this idea by purifying the stem cell and activating the different frizzles. And our hypothesis was that only activation of Frizzled-5, which we saw in our single cell data, should enable the stem cells to grow. And this is what this looks like. So you can see here, when we isolate these cells, they don't really grow without Wnt. You can give them Wnt, and then you can start to see these little things here. So this is the growth. If you try to give the Frizzled-1 or the Frizzled-2 activator, nothing happens, which is consistent with our hypothesis because these receptors are not on the surface of the cell. And only when you give Frizzled-5, which is present, do you see the outgrowth of these stem cells. They form these structurals, which we call organoids. In fact, the engineered Frizzled-5 agonist actually works better than the Wnt ligand by quite a bit, maybe by about 5 to 10-fold, which is really amazing when you think about the fact that this is a synthetic molecule. Okay, so that's in a dish. What about a more clinically relevant animal model? Can we boost stem cell activity without exacerbating fibrosis? And so we want to test this idea. We gave mice a high dose of bleomycin, which is known to cause tissue scarring and initiate cell repair. And then we injected the Frizzled-5 activator that I told you about. And it was very gratifying to see that indeed, when you inject this Frizzled-5 activator, the ability or capacity of the stem cells to regenerate the tissue is boosted by about 500 to 600%. And crucially, we can do this without exacerbating the fibrosis, which was a major goal of this field. And ultimately, this increases the survival of the mice by about 50%. So what I showed you today is that by understanding the basic biology of how our cells talk to each other, we can find therapeutic opportunities for seemingly intractable problems. And the study was exciting for me because I've worked in this field for about a decade, and this really was the first proof of concept, showing we can actually use Wnt to increase the capacity of these cells by sending them a molecular signal that we created. And lastly, the same scenario that I described to you in the lung, specifically in the alveolus, can be found in a variety of tissues where there are Wnt-dependent stem cells that we want to boost, but Wnt's also boosting the scar tissue. So now that we can have this information, we can think about achieving optimal outcomes in disease where we can both activate Frizzled-5 while inhibiting simultaneously Frizzled-1 to really sort of profoundly change the capacity of the organ to repair itself. So with that, I'll stop and thank you for your time and thank my mentors, and I'll take any questions.
All right. Thank you, Ahmad. That was actually very beautiful. So there's a few questions coming through, but I wanted to highlight, you started your talk with this beautiful little organism, this axolotl that can regenerate very well. Are the principles that you're finding in the lung, are they going to be conserved at that regenerating organism there of axolotl as well?
Yeah, actually, what's interesting is it's not always going to be conserved, but very often and very surprisingly, it is. And there's one incredible study where it's actually not the axolotl, it's the zebrafish and in the eye, but it's a similar idea where if you activate the signal that comes on in the regenerative organism exactly at the same time, you can get the mammalian cell, which is ours, to actually behave like the cell of the zebrafish in this case. So you can really actually, in some cases, do a like for like swap across hundreds of millions of years of evolution, maybe. I don't know if that's hundreds of millions, maybe tens of millions of years.
That's a lot. That's a lot.
Don't quote me on that, but yeah, it still works
Yeah,
in many
that's
cases.
fascinating. So also we have a few other questions coming through. So when you're giving these frizzled mimetics, I'm assuming that because of their proteins, you're having to inhale them, or how are they actually getting them into the mice, or how do you envision this actually going in humans? Would they inhale the therapeutic, or would it be an IV injection? What are you thinking?
Yeah, so in the mice, we inject them systematically, and incredibly, the mouse tolerates it quite well. Um, in a human, ideally, we would do exactly what you're suggesting, which is we would send it as an inhaled therapy. And this has been successful for other pathways, for example, like the inhaled JAK inhibitor, I believe has made it through clinical trials. And so I think you can certainly achieve a high level precision because you can get with Frizzled 5 the sort of intra-organ specificity. And then with the inhaled approach you highlight, you can get the intra-organ specificity, which should be the safest way forward.
That's really fascinating. So we have a question coming through. Does your Wnt mimetic cause polarity in the stem cells and induce asymmetric cell divisions? This is a highly technical question.
Yeah, yeah. So we were really actually worried about the polarity complex. And we don't see any sort of change to the polarity machinery when we stain for it. We can even, I didn't show it today, but you can even target clearly this person is a Wnt aficionado. And you can actually even target the receptors that are involved in frizzled polarity and get them to work without disrupting the polarity machinery within the cell.
Fascinating. So this is probably on everyone's mind, but one question is, what about conditions like long COVID? Do you think that there's going to be a way that this could be used to treat long COVID?
I think, it depends on what exactly is the issue in these problems. So if fibrosis comes to be seen as a big component of long COVID, then I think there is a lot of promise there. So it depends on understanding exactly, or if perhaps the stem cells have ceased function, then I think there is a lot of hope there for this type of approach of rehabilitating the stem cells. But probably some more basic biology to light the way there would be helpful.
And I know that you're, always worried about, assessing the risk of your treatments. And some of the questions coming through are, could some of your agonists actually drive cancer? Do you have to have this balancing act that you're going to, you had an inhibitor with an activator? Is that the way it's going to have to work out, or you think you can use one and not the other?
Yeah, yeah. So I get this cancer question a lot, and I used to have a sort of I would say a tongue-in-cheek answer, but turned out to actually be a true answer, which is that cancer is driven by the selection of for mutations that basically activate these pathways, and by giving these agonists or these activators, you're taking away that selection pressure.
Mhm.
And so again, I used to say this as like a conceptual answer, but it turns out that a lot of patients take lithium chloride, which is a Wnt agonist.
Mhm.
And what's interesting is that those patients are protected from cancers that are driven by Wnt, specifically like colorectal cancer. So I think there are people out there who are actually promoting the idea of using these as anti-cancer therapies.
That's really exciting. So this is, I mean, I think your research is highlighting, the fundamental basic mechanism of how the signaling system has some modalities that can be utilized in different tissues for different types of regeneration. And so there's a lot of buzz, out there. I got to ask you the questions, how far out in clinical trials are we, do you think, applying the
Yeah, so
science?
it's modulating Wnt has been a difficult clinical um, uh, avenue for a decade or so. But actually, recently there was a big success in a phase 1 clinical trial by a company called iBio, where they were injecting very similar molecules. That just the only difference is that they targeted a different frizzled receptor, and they were really, of course, it's a phase 1 clinical trial, but they saw some really strong effects in diseases like retinopathies. And so I think we're not actually that far in the next 5 or 10 years of seeing these things go to the clinic. However, as you go from the eye, which again has a very localized administration route, to something more systematic, it's going to be, I think, the toxicity will have to be explored more rigorously there.
Yeah, I really, really appreciate all of your work. And looking at the caution, throwing caution in there as well is always super important. But it sounds like you're on the cusp of finding something that may be universal for many different types of regeneration. So Ahmad, I want to thank you again. This has been an absolutely fascinating discovery, and thanks for sharing it with us. Those of you that want to ask more questions, we will at the end have some more time as well. So please definitely feel free to put in more questions for Ahmad. We can get to them at the end. But now we're going to move on to our next speaker of the evening, and this is my colleague Sarah Stanley. She's an associate professor with joint appointments in the Molecular and Cell Biology Division of Immunology and Molecular Medicine, as well as the School of Public Health in the Division of Infectious Disease and Vaccinology. So Sarah's lab studies the bacterial pathogen that causes tuberculosis, one of the deadliest and most common infectious diseases in the world. Billions of people have become hosts to the bacteria, a large reservoir of latent disease that threatens global health even further. Professor Stanley and her student researchers seek to understand why natural immunity is not more effective, what types of immune responses are protective, and how to use this knowledge to develop new vaccines and therapeutics for pathogens of global significance. Sarah is truly going after the unmet. Sarah recently received a 2025 Distinguished Teaching Award that we highlighted earlier. This is one of our campus's highest honor for teaching, and only 3 or 4 of them are given out every year amongst all the faculty. So it's a real pleasure that Sarah was recognized for her work. She teaches not only at UC Berkeley, but she also volunteers some of her time during the summers to teach in Africa as well. So Sarah, thank you for joining us this evening. Can you share your research with us?
Yes, thank you so much, Andy, for that really nice introduction. And I'm very excited to talk about the disease that I work on, which, as Andy introduced, is tuberculosis. There's probably some of you in the audience who are thinking, oh, tuberculosis, I've heard of that, but isn't that a disease, that's kind of a historical thing? And in many ways, that's true. And TB is thought to be one of the oldest of all human infections. It emerged probably around 70,000, more than 70,000 years ago, and has been infecting and co-evolving with humans ever since. The peak of TB in some ways was in the 1800s and early 1900s, when it was estimated that 1 in every 4 deaths in TB and Western Europe was caused, in the US and Western Europe was caused by TB. The first antibiotic for TB was introduced in 1944. And although the introduction of antibiotics and a vaccine, which we now know is questionably effective, allowed us to gain control of TB in the wealthier parts of the world, in much of the world, TB is still a major cause of disease. And in fact, in 2023, it was estimated that TB killed 1.5 million people across the world. So TB is still a major problem worldwide. And one of the reasons for this is that surprisingly, there's a lot about the biology of disease that we still don't understand. So TB is a disease of the lungs, and like many respiratory diseases, it's spread when an infected person coughs and they cough out bacteria, and those bacteria are taken up into the lungs of an uninfected person. The first cell that this bacteria encounters is called a macrophage. So a macrophage is a cell that is designed to take up bacteria or other foreign material and kill it. And so in the case of TB, whether macrophages are able to kill the bacteria really determines the outcome. So if macrophages take up the bacteria and are able to kill them, the bacteria is not able to cause disease, we have controlled infection, and the person is healthy. On the other hand, if macrophages are unable to kill the bacteria actually use the macrophage as its niche for replication. Bacteria will grow inside the macrophages, ultimately kill the macrophage that it initially infected, spread to new macrophages. And as the immune response tries to keep up and tries to kill the bacteria, inflammation increases in the lungs. And it's this immune response that actually ends up causing tissue destruction and the symptoms of the disease tuberculosis, which include coughing, hemorrhaging from the lungs, weight loss, fever, etc. So surprisingly, we still don't understand why is it that in some people, TB can enter into the lungs, the macrophages are able to control the infection, and the person does not develop tuberculosis, whereas in other people, the macrophages fail and symptoms develop. So I'm going to tell you about a new discovery that we have that relates to this outcome of whether or not a person is able to control infection with TB. But I'm going to seemingly take a completely 180 and tell you about something that is seemingly completely unrelated, but I promise I'll come back to TB in just a moment. So I need to introduce you to a mutation in a gene called ALDH2 or aldehyde dehydrogenase 2. And this mutation is the most common mutation in humans. So this mutation in the gene, it causes the protein that's encoded by the gene to be non-functional. And what I'm showing you in this map is pie charts of different populations around the world, looking at whether a person has the wild type, so the unmutated version of this gene, that's shown in yellow, or a mutated version of this gene, and that's shown in green. And what I hope you can appreciate is that in parts of the world, including East Asia and Southeast Asia, there are relatively high rates of this mutant version of ALDH2 in populations. And why this is has been a mystery because this mutation is associated with some negative health consequences, including elevated risk of cardiovascular diseases and some cancer. So why does this mutation exist? You've probably heard of this gene, or you at least know what it does, even if you're not aware of the name of this gene, because ALDH2 is most famous for its role in detoxification of ethanol. So when a person drinks, of course, ethanol is toxic. It needs to be detoxified. And the way that is accomplished is first by an enzyme called alcohol dehydrogenase that takes ethanol and it converts it into a molecule called an aldehyde. Aldehydes are highly toxic. Actually, this aldehyde is much more toxic than ethanol. And so this molecule needs to be converted to a harmless compound. And the protein that does that is called ALDH2, the gene that we've been discussing. If a person has a non-functional ALDH2, what happens when they drink alcohol is that this toxic aldehyde intermediate builds up. These aldehydes are highly reactive. They can react with DNA, they can react with proteins, they can react with the surfaces of your cells. And so when a person with this mutation drinks alcohol, we see this characteristic flush that is a result of the toxicity of this aldehyde on the vasculature. Okay, so what does this have to do with TB? Well, it turns out that macrophages are known when they ingest bacteria to activate programs that generate the production of aldehydes as byproducts. And some of these aldehydes are known to actually be detoxified by ALDH2. So we developed the hypothesis that maybe in people who have this ALDH2 mutation, when their macrophages get infected, they make aldehyde, they're unable to detoxify this aldehyde, and because this aldehyde is toxic, it can actually inhibit the growth of the bacteria in the cells or even kill the bacteria. So how do we test a hypothesis like this? The first thing that we wanted to do was we wanted to know, is this true in human populations, that people who have this mutation are protected against TB infection? So to ask this question, we collaborated with an epidemiologist at the University of Melbourne Her name is Sarah Dunstan, and she follows two cohorts of TB patients, one in Vietnam and the other in Singapore. And these TB patients either have pulmonary tuberculosis, so TB in the lungs, or they have TB meningitis, which is TB in the central nervous system. And when she looked at these patients and looked at whether or not they were likely to have tuberculosis, or whether they had tuberculosis, and whether they had the wild type or the mutated version of ALDH2, what they found was that people with the mutated version of ALDH2 have a 20% lower risk of having pulmonary tuberculosis. Interestingly, however, there was no change in the risk of having tuberculosis meningitis. So this is really interesting, and it supports our hypothesis that ALDH2 mutation can protect someone against infection with tuberculosis. But one of the challenges of doing research with humans is that, humans are out there, they're drinking, they're living lives, they have very diverse genetics. And so how do we really know that this result is because these humans have some intrinsic ability to be more protected against TB infection, or they're just healthier because they don't drink? So the way that my lab addresses this is using mouse models. So I'm going to show you one experiment that we've done, which is that we took mice. So we took either the wild type version of the mouse, or we made a mouse that has a mutation in ALDH2 that mimics the human mutation. We took these mice, we infected them with tuberculosis, we waited for a month, we then harvested the lungs from these mice and we counted the bacteria that were in the lungs. So in infection of the wild type mouse, what we see is on average, we see 10 to the 8th, so almost 100 million bacteria in the lungs of these mice. Really exciting was that when we looked in the lungs of mice that had a mutation in ALDH2, we saw on average 10-fold fewer bacteria in the lungs, indicating that this mutation is able to give some degree of protection against TB infection to these animals. And really supporting our hypothesis that ALDH2 and the aldehydes that ALDH2 generates can help determine the outcome of whether macrophages are able to control infection. So we have a lot more data that I'll just summarize here in the interest of time. I told you that mutation of ALDH2 provides an advantage against infection with MTB. And what's really exciting is that we found that this is also true against other intracellular bacteria. And in fact, for some bacteria, we see an even more dramatic effect of having ALDH2 conferring protection. The protection that we see seems to be specific to the lung. And this is something we don't understand, but when we look at other tissues, they don't seem to be as protected by this mutation as the lung is. So one thing we're really excited about is, can we harness this pathway for the development of therapeutics? For people who have a wild type allele of ALDH2, is there a drug we could give them that would mimic this mutation? And another thing that I think is really exciting is that this is really just the tip of the iceberg. What we're learning is that there are other aldehydes that are generated by macrophages and actually detoxified by other enzymes independent of ALDH2 that seem to also contribute to control of bacterial infection. So what we think we've discovered through this work is an entirely new arm of the immune system and a new way that macrophages can kill intracellular bacteria like TB. So with that, I will thank the people who did this work, my postdoc, Sam Berry, my student, Scott, all the rest of my lab, and all my really wonderful collaborators here at Berkeley and then across the world. And thank you all for your attention.
Really exciting, Sarah. It's really fascinating. I couldn't help but think about the parallels that have happened, with sickle cell, the alleles there of hemoglobin and protection against malaria. It's a deadly disease, but if you're just mutant enough, you can actually be protected from malaria. It seems like LDH2 may be something similar that may be a little bit bad for you, but it's going to really protect you against TB.
Yeah, that's exactly how we're thinking about it, is the parallel that we use is that sickle cell is protective against malaria. And we think that ALDH2 is protective against TB, but also against other bacteria as well. So it's going to be the work of time to figure out what is actually the infection that has driven selection in humans, and maybe it's a multifaceted answer. But yeah, we're really excited about that.
That's really, really fast. So we have some other questions coming through. So these macrophages, which are the first line sort of defense for the lung for infections, they're building up this toxic molecule, this aldehyde. That must be bad for the macrophage. Do they get regenerated? You know, going back to Ahmad's talk, what's the system there to make sure that we still have enough of these macrophages when they're building up this toxic defense mechanism?
Yeah, that's a great question. And we were actually really surprised when I didn't show you this data, but we've analyzed macrophages just in a dish from these mice. And we see that these macrophages from the ALDH2 knockout mice do make more aldehyde. And we thought that that would kill the macrophages, but in fact, it doesn't. And
Oh.
so what we think is that that mammalian cells are actually just much better at repairing the damage caused by the aldehydes than bacteria are. So the macrophages can afford to upregulate production of this toxic molecule and survive it, whereas it can be used to kill the bacteria. And there's actually other things that are known about how macrophages kill bacteria that are kind of analogous. So, for example, one of the things macrophages do to kill bacteria, not TB, but other bacteria, is they make hydrogen peroxide, like the molecule that you can buy at the drugstore and put on your cuts or whatever. Macrophages actually produce that to kill bacteria. TB is hardy, so it's not killed by hydrogen peroxide.
I hope I have really good macrophages. So one quick question. Is there a connection between TB and the fibrosis that Ahmad's looking at? Does TB actually develop a fibrotic lung in the late stages?
Yeah, I'm really glad you asked that question because the answer is there is. So TB is a really devastating disease. If you're not treated or if you have drug-resistant TB, then of course the infection itself can cause mortality. But even in people who are successfully treated with antibiotics, they often have long-term damage to the lungs, similar probably in some ways to what is seen in COVID, in long COVID. And one of the things that can happen to these patients is that they have fibrosis, and this increases their risk of mortality by quite a lot, actually, and contributes to risk of death in many parts of the world is post-tuberculosis fibrosis. And so actually Ahmad and I have started talking about a collaboration to try to see if we can use some of his tools in the context of infectious disease as well.
Awesome. Well, Sarah, thank you again. It's really fascinating. You are going after the diseases, one of the major diseases that pharma is not going to go into Africa and try to go after. So it's really fascinating that you're going after this and making inroads and finding these amazing discoveries. So thank you again, Sarah. And there'll be more questions at the end available for Sarah. So please put them in the chat and we can get into those as well. So I think it's time for us to move on to our next speaker, who is Diana Bautista, who is a Howard Hughes Medical Institute investigator, as well as a professor in cell biology, developmental biology, and physiology. So her lab aims to decipher the neuroimmune interactions driving chronic itch, pain, and airway inflammation. She has some of the best videos we're going to see all night of mice actually sneezing. It is truly fascinating, some of the research that she's doing, looking at this neuronal response to get us to sneeze. So her team is investigating the molecular and cellular mechanisms by which sensory neurons, airway epithelial cells, and immune cell interactions become dysregulated following injury, atopic disease, and infection. Their work involves studying clinically relevant disease models of skin and airway inflammation, including human and mouse models of airway inflammation and respiratory stress caused by inflammatory mediators and viral infections that I think we are all very familiar with. So Diana, thank you so much for joining us, and please take it away and tell us about some of your research.
Great, thanks, Andy. Let's see if I can share this. And great, how's that look? Good. All right, well, I'm really excited to share some of our work today looking at neural mechanisms of SARS-CoV-2 infection. For any organism to survive, it needs to be able to protect itself from a range of threats. These could include toxins or pathogens in our environment, noxious mechanical or thermal insult, and a variety of other irritants. And metazoans have evolved three key systems for protection. First, there are epithelial cells that form a very important barrier between an organism and its environment. Second is the immune system that performs surveillance and clearance of pathogens and other harmful elements that enter the body. And lastly, sensory neurons that innervate densely throughout our body drive protective physiological responses. And my lab studies the molecular and cellular mechanisms by which these sensory neurons promote protective responses to a variety of harmful threats. So for example, we showed that avoidance to noxious cold that you could see here in this video on the left, that this mouse is avoiding the left side of the chamber that's cold, is dependent on a sensory neuron-specific cold and menthol-sensitive ion channel called TRPM8. In the video on the right, mice lacking this neuronal ion channel do not avoid the cold, and they spend their time equally on both the cold and the warm plate. And more importantly, these animals lacking TRPM8 don't develop cold pain hypersensitivity under a variety of disease states. And we now know that there are a variety of SNPs that have been identified in people that's correlated with the ability to develop migraines and migraines with aura. So we're very interested in this TRPM8 ion channel in the context of thermosensation, but also pain disease states. Now, another type of protective response in the mammalian airways includes sneezing and coughing. These reflexes help prevent insult to the lungs, which of course we know from our other speakers is vital for breathing. And as you can appreciate in this video, a sneeze triggers a vigorous expulsion to clear an irritant from the nose and the upper airways. And it's similar with cough for the lower airways. And it's thought that this might protect damage by getting rid of whatever that irritant or pathogen is. Now, sneezing is also one of the most common symptoms of respiratory infections, and we've recognized its ability to spread disease for a long time, as you could see here in this public health poster from the 1940s. And even 80 years later, during the COVID-19 pandemic, one of the most effective approaches to limiting viral transmission has been wearing a mask. Yet, to date, we still know very little about the cells and molecules that mediate sneeze or cough, or even promote respiratory distress during viral infection. So my lab is very interested in understanding how these sensory neurons that densely innervate the head and upper airways mediate sneeze and headache and other types of viral invoked symptoms. And we're also interested in the sensory neurons that densely innervate the body and the lungs that regulate breathing, drive reflexes like cough, and initiate pain sensations that you may have also experienced the last time you were sick. Sore throat, aches and pains, and that general feel of discomfort. But we really don't know how these sensory neurons are activated. We also know that post-infection, that these same sensory neurons mediate many symptoms of long COVID, persistent changes in breathing, joint pain, chest pain, and persistent cough. So we want to try to understand how viral infection activates sensory neurons to drive these symptoms of infection. And we know that these sensory neurons in people and in a wide range of animals can be directly infected. But we also recently started to ask about whether viral proteins can also directly activate sensory neurons during early infection of the airway epithelium. Now we know that SARS-CoV-2 genome encodes a variety of proteins, and the first proteins that are translated are proteases, and there's two of them called papain-like protease and the main protease. These proteases play a key role in processing all of the subsequent viral proteins needed for viral replication. And because of this role, they've been of much interest as therapeutic targets for COVID-19. And if any of you have taken Paxlovid when you were infected like me, the Mpro protease is the target of Paxlovid. Now, under a variety of different disease states in the airways, including asthma and COPD and other respiratory diseases, it's long been known that elevated proteases are a key instigator of airway dysfunction. And we and others have shown that these proteases can directly act on sensory neurons to drive changes in breathing and defensive reflexes. So a very talented student in my lab, Sonali Mali, asked if SARS-CoV-2 proteases could directly activate sensory neurons. To test this, we performed brain imaging in an anesthetized mouse and asked if we put a small drop of protease into the nasal cavity, if this activates sensory neurons. And in this experiment, we can look at hundreds of sensory neurons simultaneously to ask if they are activated. And in this video, you could see these flashes of light in these small round cells that you could see here. And what you could see is that when we perfuse in the protease, we see a subset of cells activated. And this image just highlights three of those cells. And we could quantify here on the right in this graph, and we look at neuronal activation as a function of time. And you could see that there's these peaks of activation very quickly after a small drop of this protease goes into the nose. Okay, so what are the consequences of this? So as Andy mentioned and named, we developed a sneezometer by combining high-speed audio recordings with high-speed video recordings and adding a blue dye into the nose so that we could see the actual secretions that come out of the nose. And I'll show you a video highlighting this. First, I'll show it at normal speed, and then I'll slow it down so hopefully you could see and possibly even hear the result. Here's in slow speed. Here you can see the wall is white. Hopefully you heard that roar of a sneeze or a chew, and you could see the expulsion. And we could quantify this in a number of ways. We could quantify how much dye comes out and splatters the wall. We could look at frequency of sound and video movements in the high-speed video recordings. And this just is a quantification here where each of these rasters is a sneeze, each row is a different mouse, and you could see that the protease evokes a lot of sneezing, about 1 every 10 seconds over a 2-minute period, whereas just putting in dye alone triggers a couple of sneezes, but not much discomfort. I don't have time to show you the data, but we identified a key ion channel in these sensory neurons related to the TRPM8 cold sensor called TRPA1. Mice lacking this receptor do not sneeze or cough in response to proteases. And I didn't show you this data either, but we also see that a brief exposure to protease can lead to prolonged pain hypersensitivity that's also dependent on TRPA1. And we think that this is important clinically, as we currently have no effective treatments for acute or persistent viral cough. And there's a TRPA1 antagonist that's currently in a phase 2 clinical trial right now that we think might be effective for treating a variety of airway dysfunctions related to sensory neuron activity under viral infections. And of course, we're looking more broadly to look at the role of sensory neurons in SARS-CoV-2 infection models, to look at changes in respiratory function, pain hypersensitivity, neuroinflammation, and also viral headache. So with that, I'll just thank the people who did the lab, and thank you so much for taking the time to come out tonight.
Very fascinating, Diana. So you're at Berkeley now, and you've got to see videos of mice sneezing and actually have it quantitated. So I know there's a ton of questions coming out. It's really beautiful work. So do you think, you mentioned that there's these inhibitors that are in clinical trials to inhibit this ion channel that's in the sensory neurons. Do you think that that will make people more sensitive or more resistant to COVID? So if they can't sneeze, will they become more infected? Or how, what's the chicken and the egg?
Yeah, it's a great question. We think about it a lot. Is sneezing something that evolved to help us clear the virus before it gets deep into the lung or has more time to replicate? Or is it something that the virus has co-opted to trigger sneeze and viral spread? And of course, we don't really know, but we hope that some of our future studies looking at some of these roles of sensory neurons in different aspects of infection could shed light on this really interesting question.
Yeah, I think you have everything set up to address it. So you, in the beginning of your talk, I mean, it's so encompassing what you're studying is, the cells that protect the organism, the epithelial layer, and then you have the sensory neurons and the immune system. So when you activate the sensory system, do you see immune cell activation happening? Is this something that you predict as well?
Yeah, so we now know it was previously thought that when you get infected, the epithelial cells get activated, immune cells come, and then they passively, activate these sensory neurons that drive these reflexes. And now we know that that's not really the case. That sensory neurons are playing a key role in disease pathogenesis, and that they have the ability to manipulate the immune system. In some cases to stop it, in other cases to activate it and to call in different cells depending on the disease state. And so this is something we're actively studying with SARS-CoV-2.
Yeah, I think you're one of the very few people in the world that's, making the marriage between neuroscience and immunology, really bringing these two fields together. It's extremely fascinating. Let me look through and see what other questions we have going on here. So when you move into the cough system, do you think it's going to be the same sensory neurons or the same channels that are going to be doing cough? You may have mentioned that a little bit, but what do
Yeah, we think that when they're, first of all, somatosensory neurons are very diverse, and we've identified one specific subset that triggers this sneeze cough cycle. But we think that there are others that get activated, and we know that there are other viral proteins like MPRO that I mentioned early on that activates different subsets. So we think that some might be mediating these airway defenses, others might be promoting headache and aches and pains, and we're working to dissect that out.
That's really fascinating. All right, Diana, thank you so much. I think we're going to bring all the speakers back in and tie everything back together and get some more questions from the rest of the audience. And so if Sarah and Ahmad can come back in. Really fascinating talks. It's, bringing everything together here of lung biology and sensory responses to it and infectious responses to it. So there was a question here actually for Sarah that I would be fascinated for you to expand on because you do so much work, not only at Berkeley, but also in Africa as well. Can you highlight some of those sort of broadly, some of the things you've done of going out in the field and doing some of the things that you've done and also some of your educational things you've been doing as well?
Yeah, I mean, I could talk all night about this, but I'll be brief. So, I have two sort of activities. One is through a organization on campus called the Alliance for Global Health and Science. We do scientific capacity strengthening in East Africa. So what we do is we go to universities in East Africa. We ask them what their barriers are to be able to do research on the diseases that affect them. What kinds of skills do they need? What kind of equipment do they need? How can we help promote their capacity to do independent research? And so that's one of the major programs that I run in East Africa. I also do research there. So I have some kind of smaller and newer projects around trying to diagnose tuberculosis in children. We're talking about the lung. Actually, the way that you diagnose tuberculosis is you cough and you bring up sputum, but children don't bring up sputum, and so they're very difficult to diagnose. And so I'm a very small part of a much bigger study trying to identify new diagnostics in kids.
Okay, we have someone wanting to raise their hand, but we have them muted. So Ted Crumlin, can you please type your question into the chat? Okay, we have Dean Harland's gonna, in Ahmad's work on the specificity response, how much is this driven by the different Wnt molecules?
Yes, I'm unmuted so people can hear me. I think the Wnt molecules clearly play. Some of it is driven by the specificity of the Wnt molecules because there are 19 of them, and if you knock out different Wnt molecules, you get different phenotypes in the body. So, for example, if you knock out Wnt2 or you get lung agenesis, which basically means there is no lung. So clearly, the specificity of each molecule is quite important. However, the genetics has been challenging, and following study. So the genetics is challenging because there are 20 molecules to knock out. You have to knock them out in a specific cell type. So it's been slow going. But the other thing that's kind of interesting is studying a molecule being sent and going many places is harder than studying the receiving molecule that just sits on one cell. So I think for that reason, the focus has been on the receptors, despite the pathway being named after the ligand. Yeah, but certainly some level of molecule specificity plays a role in the cell type effects that we see.
Yeah, I think it's going to be, you're laying down the foundation for many different molecules interact with each other to regenerate in many different contexts. And so it's going to be exciting to see how you unravel this, going forward in your career. The example you've given already is complex enough, but it seems like it's going to get even more interesting and exciting as you move into other tissues that are regenerating as well. Let's see if we have some more questions here, going through some of these things. So, Diana, are there actually vertebrate species that don't sneeze? Or does every animal have the capability to sneeze?
Well, it's interesting that you asked that question. For a long time, there's been a controversy about whether mice actually sneeze or cough. And it's a hot topic in biology right now in trying to figure this out. And so we and other people have started really digging into this. And I'm convinced that mice do sneeze and that they do cough. But there's been a lot of work done to really prove that, looking at the whole body physiology to really prove that. And I think that opens up a lot of opportunities for preclinical studies using mice and taking advantage of the great molecular genetic tools to look for new therapeutics and new genes and molecules and cells that regulate it.
Yeah, I would like to have my trip A sequence to see if I'm hyperactive, because when allergy season happens, I'm just constantly sneezing one after time after time after time. So maybe I could be one of your participants in that study. Okay, so we're getting questions here. What portion of this invaluable work is supported by federal grants? This is a timely question, I should say. I would say probably almost all of it, amongst all of us, is supported by federal grants. So great question, Ted, and we're waiting to see how this is going to be, how we're going to be affected by it down the road. So we have one more question for Professor Stanley. Can your findings about the ALDH2 gene be applied to other conditions besides TB? I think you mentioned there were some other bacterial infections. You didn't name them, but maybe you could now on some other things that may be useful.
Yeah, so there are other infections. I'm trying to think of one that people here would have heard of that we've tested. So one of the ones that we see protection is methicillin-resistant Staphylococcus aureus, which is a skin infection that I think you hear about a lot in the news, and it's also relatively common in people. So we see that this mutation seems to protect against infection. The caveat to that is that's not in the skin model, but in the lung model of infection with staph. We've tested a number of different bacterial pathogens. We've really focused on ones that are for the lungs. Many of them are just kind of ones that we work with in the lab, and most people haven't heard of. They're kind of model organisms, but we're really interested in moving into some of the more clinically relevant infection, causes of infection. And one area that we have not gone is the gastrointestinal system. So we're really interested in testing those pathogens as well. Great question.
Yeah, great question. Great question to our audience and participation. I'm afraid we're out of time at this point, and so we're running a little bit close to time, so I'm going to hand it back over to Dean Harland.
Thanks, Andy, and thanks for moderating, and thanks to all the speakers for their amazing presentations. I think we can all now go and tell our friends that we in the University of California, Berkeley are helping the society through our fundamental research. And these are talks that particularly clearly are going to benefit us in the long run. I hope you'll join us not next Thursday, but Thursday, April 17th at 5 o'clock for the next installment of Basic Science Lights the Way. There we're going to hear from 3 physical scientists about what their research has revealed about our solar system's largest planet. I think we all know who that is. From the fluid dynamics of the Great Red Spot to the mysterious magnetic tornadoes at its poles. Further, if you'd like to learn more about anything we covered, please reach out to us. The video of today's session will be available at the Basic Science Lights the Way website about 2 weeks from now. But in the meantime, you can catch up on previous episodes and watch the episodes that you've missed. So thanks again for attending and showing such interest in this topic and in basic science in general. We couldn't do our work without your interest, your attention, and your support. So until next time, stay curious and fiat lux.